Hello, and welcome everyone to Bio-Rad's webinar series on Droplet Digital PCR. This is our 6th webinar Bio-Rad is hosting on ddPCR. My name is Mohini Patil, and I'm the Global Product Manager for Droplet Digital PCR at Bio-Rad and your host for today's webinar. First, let's discuss the webinar logistics. The webinar will last approximately 1 hour. Dr. Jack Regan will present on ddPCR and its application in phasing of genetic variants for about 45 minutes. Following the presentation, we will have 15 minutes of live questions and answers with Jack. Throughout the webinar, feel free to use the Q&A box on the left-hand side of your screen in order to submit your ddPCR questions directly to us. We will get to as many of your questions as we have time for. However, if we cannot get to your question today, we will personally follow up with you over the next few weeks. Also, if you have any technical difficulties during the webinar, please use the Q&A box to report them. Now, a little bit about our speaker, Dr. Jack Regan. Dr. Regan earned his PhD from the University of California, San Francisco, where he studied the influenza polymerase. Jack completed his postdoctoral studies at Lawrence Livermore National Laboratory. At Bio-Rad, Jack leads a team of scientists who focus on new application development. The title of Dr. Regan's talk this morning is Phasing of Genetic Variants for CFTR Haplotyping. Without further ado, let's get started. Jack?
Thank you for the introduction, Mohini. Today I will describe how ddPCR can be used to phase variants. After introducing the methodology, I will present data on phasing variants found in cell lines derived from cystic fibrosis patients. Knowing the phase of variants for this disease, as well as many other diseases, is necessary for making informed medical decisions and for genetic counseling. First, I have one slide in which I will review the instrumentation and workflow for ddPCR. Simply put, ddPCR is a methodology that allows for direct, absolute quantification of nucleic acids in a sample. This is achieved by emulsifying a standard TaqMan reaction mixture into thousands of droplets, which are approximately 1 nanoliter in volume. These droplets are PCR amplified to endpoint in a thermocycler and then analyzed for fluorescence in a 2-color droplet reader. The droplets are classified as either positive or negative based on a fluorescence threshold. The fractional abundance of positive droplets is used to determine the number of target molecules present in the reaction mixture. The precision and sensitivity of ddPCR benefit many applications, including absolute quantification, rare mutation detection, copy number determination, and phasing. If you are interested in learning about more of the first 3 applications, please view our other ddPCR webinars using the provided information. So, what exactly is phasing? Phasing, also called haplotyping, is resolving the specific arrangement of variants or alleles along a stretch of DNA. The arrangement of variants or alleles only comes into question if the genome being queried has 2 or more copies. In other words, the sample must have at least a diploid genome. Here, I've drawn 2 autosomes of a cell, where one chromosome was inherited from the father and the other was inherited from the mother. If this sample were to be sequenced, we would learn that the locus is heterozygous for both the A and B locus. However, we would likely not learn the phase of the discovered variants. This can be problematic since adjacent variants can affect one another, and knowing their arrangement is important for diagnosing and understanding the causes of diseases. For this presentation, I will depict variants in lowercase lettering and wild-type sequence in uppercase lettering. In this illustration, the lowercase a variant is on the same chromosome as the lowercase b variant. When variants are on the same stretch of DNA, they are considered to be cis-configured. Alternatively, if one of the variants was on the maternal chromosome, these variants would be trans-configured. There are several obvious applications that benefit ddPCR phasing. First, for genetic counseling and clinical interpretation, it is very important to be able to distinguish between disease carriers and compound heterozygotes. This is particularly true for diseases such as cystic fibrosis, Accordingly, this presentation focuses on this disease and how ddPCR can be used to resolve two samples of identical genotype that might have significantly different phenotypes due to the phase of the variants. A second application is understanding allele-specific expression, and a third is mapping genomic structure. Today, I will be presenting ddPCR as a tool to make advances in these applications. In the next few slides, I will cover the principle behind using ddPCR to determine whether variants are cis or trans configured. Here, I show two independent DNA molecules. In this presentation, I will represent DNA molecules as straight horizontal lines and depict the targets of interest with vertical lines, accompanied by lower and/or uppercase letters to indicate whether the targets are variants or wild-type sequence, respectively. Here, I show two variants. As I previously mentioned, ddPCR uses TaqMan assays. Here, a FAM assay, shown in blue, is specific for the A variant, and a hex assay, shown in green, is specific for the B variant. If we were to analyze this sample using ddPCR and view these data in a fluorescence scatter plot, we would see four distinct clusters of droplets. In this plot, each circle represents a cluster of droplets. Each cluster may have anywhere from a single droplet to thousands of droplets. At high enough DNA input levels, we expect some A and B molecules to co-localize due to chance. This creates the double positive cluster shown in orange. Now let's pretend that the two targets are arranged along a stretch of DNA and that some of the DNA is fragmented. For the fragmented samples, A is separated from B. So the DNA molecules that are still intact, A and B, are physically linked to each other by the intervening DNA. In phasing, we are looking to measure these intact molecules. Therefore, we must take care not to accidentally separate these two targets. Accordingly, for phasing applications, we generally skip the restriction enzyme digestion step, which is a recommended step for other ddPCR applications. When analyzing a sample that has intact DNA molecules containing both A and B variants, we will observe more double positive droplets than we would expect due solely to chance colocalization of two independent variants. This increase is due to the presence of the linked molecules. If the increase in the double positive droplets is beyond what we would expect from statistical variation, then we can claim that the two variants are linked. In other words, these variants are part of the same haplotype. Here, I've added an actual fluorescence scatter plot to emphasize that we are looking at populations of droplets. And here is the scatter plot without the identifiers. You can clearly see that there are more double positive droplets than single positive droplets. Before moving on, I'd like to mention that ddPCR phasing does not require long-range PCR, which can be technically challenging. Instead, it relies on two distinct small amplicon-sized PCR reactions, which only need to be efficient enough to distinguish positive from negative droplets after 40 cycles of thermocycling. Bio-Rad's QX200 Droplet Digital PCR system comes with software called QuantaSoft. Here is a screenshot of one of the data analysis tabs that QuantaSoft automatically populates based on the analyzed droplets. In the upper left-hand corner is a table that includes data from the selected wells. The exportable table has 62 columns of data. Due to the size of this table, I've opted to extract some of the more relevant columns into the table shown here. In one column, I have included ellipses to represent the 15 columns of data that I've omitted for simplicity of viewing. The two shown rows represent data collected from a single duplex reaction where concentration measurements are provided for both alleles. You will notice that the concentration of allele A is 80.4 copies per microliter, whereas the concentration for allele B is slightly higher at 81.6 copies per microliter. When quantifying two targets that are expected to be of equal concentration, it is normal to have small differences in the reported concentrations of each target. Conveniently, every time a duplex reaction is run using Bio-Rad's platform, QuantaSoft automatically calculates whether linkage exists between the two targets and reports the concentration of linked molecules in the linkage column. Both concentration and linkage column values are reported in copies per microliter. In contrast to the variable concentration measurements for both alleles, only a single value for linked molecules is reported as they are connected. To calculate the percentage of linked molecules in a sample, the concentration of the linked species is divided by the average concentration of both targets, and this quotient is multiplied by 100 to get the percentage of linked molecules. When using ddPCR to determine whether two alleles are linked, it is important to always run a negative control. We advise performing another duplex reaction in parallel that includes an assay targeting the other allele. In this example, rather than targeting the lowercase b allele, the control duplex assay would include a hex assay that is specific for the uppercase b allele. By default, if the A locus is close enough to the B locus and the DNA is sufficiently intact, one of the two duplexes should show linkage and the other should not. For this sample, we did run a negative control duplex, which is shown here. You can see that there are fewer double positive droplets, suggesting that these two alleles are not linked. In this slide, I am showing a side-by-side comparison of a test and negative control duplex assay. On the left, the included assays target linked alleles, whereas on the right, the included assays target unlinked alleles. As shown here, it is often easy to observe an increase in double positive droplets for linked molecules versus unlinked molecules. Now, on to the cystic fibrosis portion of the presentation. Cystic fibrosis is an autosomal recessive disease. It is the most common life-limiting disease among people of European heritage. In the United States, approximately 27,000 individuals have cystic fibrosis, and roughly 10 million are thought to be carriers. The disease is caused by not having enough functional cystic fibrosis transmembrane conductance regulator protein, which is involved in transporting ions across epithelial membranes. The inability to transport ions affects many organs, most notably the lungs and the pancreas. Over the past couple of decades, the life expectancy of individuals with cystic fibrosis has increased substantially. Now, the average life expectancy is 37.4 years, with the most common cause of death being cardiorespiratory complications. CFTR is the gene that encodes the ion channel protein that is necessary for ion transport. In patients with cystic fibrosis, both copies of CFTR must be compromised. Almost 2,000 variants have been identified in this gene, some of which are depicted as small dashes in this UCSC browser image of the CFTR gene. To date, 127 variants have been classified as being pathogenic, meaning they reduce the functionality of the gene down to less than 10% of a non-mutated gene. The remaining variants have either no effect, have a mild phenotype, or not enough data exists to know the impact of the variant on protein function. When diagnosing cystic fibrosis from a genetic standpoint, several genotypes can be observed. The easiest genotype for predicting clinical outcome is when a patient is found to be homozygous for a particular variant, since it is clear that both copies of CFTR are compromised. In contrast, if a patient is genotyped to have two heterozygous variants, then this individual could have variants that are either trans or cis configured. This creates some ambiguity, as these haplotypes are clearly different and may result in very different disease states. To clarify some terminology, if the two variants are pathogenic and transconfigured, then the individual has cystic fibrosis, and their genome is considered to be a compound heterozygote. The other possibility is that the variants are on the same chromosome, in which case they are cis-configured. If one or more of the variants is pathogenic, then the individual is a carrier for the disease. If the variants are normally mild if found alone, but collectively the two variants become pathogenic, then this is called a complex allele, and the individual is a carrier for cystic fibrosis. In the next few slides, I will focus on a specific example of a complex allele in cystic fibrosis. A common complex allele in cystic fibrosis is when an arginine residue at position 117 is turned into a histidine residue, and this variant is cis-configured with a polythymine tract within intron 8 that has just 5 thymine nucleotides. If this arrangement exists, then improper splicing occurs and 90% of the transcripts lack exon 9, thereby compromising the functionality of the gene. Here, I've illustrated both CFTR gene copies. Say NGS has indicated that R117H and the 5T variant are present, but NGS is unable to determine the phase of these variants as they are 18 kb apart. So the question is, are these variants cis-configured, in which case the person is a carrier, or are these variants trans-configured, and the person is not a carrier? In order to confirm the haplotype of this locus, 4 unique duplex assays should be assembled to cover all possible combinations. In this illustration, the first duplex assay is specific to the arginine residue at the 117 locus and the 7T allele. The second duplex would be comprised of assays specific to the arginine residue at the 117 locus and the 5T variant. The third duplex would be comprised of assays specific to the histidine residue at 117 and the 7T allele. And the last duplex would include assays for the histidine residue at the 117 locus and the 5T variant. These 4 duplexes together can be used to conclusively determine the arrangement of the variants. From a linkage standpoint, 2 of the duplexes should show a similar level of positive linkage, and the other 2 should effectively show no linkage. We utilized this approach on a cell line derived from a cystic fibrosis patient with these variants and found it to work extremely well. We decided to expand the project to demonstrate that we could phase different variants across the gene. We ordered 6 cell lines that were positive for 2 or more variants. The approximate location of these variants along the CFTR gene are shown here. The long blue box represents the gene from the start codon to the stop codon. One cell line contained a 129 guanine to cytosine variant located in the promoter. Shown in red is the location of the polythymine tract in intron 8, which affects splicing of exon 9. The remainder of the mutations are in the coding region. I bracketed the complex allele that we just covered. In some cell lines, the variants are as close together as 11 kb, whereas others are separated by up to 116 kb. The distance separating the other variants are of intermediate lengths. We designed assays for each locus and measured linkage for all possible combinations. For the loci separated by 116 kb, we measured
18%
of the molecules to be linked. We calculated the percentage of linked molecules for all the variants across the 6 cell lines and found the distance separating the variants was a good predictor of how much DNA remained intact after sample preparation. Remember, for each pair of variants, 2 of the duplexes should show a similar level of linkage, shown in blue, and 2 should effectively show no linkage, shown in purple. The linkage measurements for each haplotype within a cell line are very similar, sometimes overlapping, as shown for the variants that are 80 kb apart, where there appears to be just one symbol, but in fact there are two overlapping symbols. At about 28 kb, there is a cluster of 8 symbols that show positive linkage and another cluster of 8 symbols that show no linkage. The 8 positive linkage measurements were split across 4 cell lines, and the difference in the percentage of linked molecules between them is only about 11%, which shows how robust and reliable this technique is for phasing variants when the samples are treated equally. Although this robustness is nice, remember that linkage is a binary measurement. Either the alleles are linked or not. The actual percentage of linked molecules is just a measure of the quality of the extracted DNA. We knew from the previous experiment that the percentage of linked molecules tracks very nicely with distance across multiple cell lines, but we want to explore how far we can measure linkage within a single cell line. To answer this question, we transitioned away from heterozygous loci and instead focused on conserved regions of the genome, so we could design assays at very specific distances away from a common anchor sequence. The targeted sequences were called mile markers. The closest mile marker was just 1 kb away from the anchor sequence. We designed other assays targeting sequences that were increasingly farther away, specifically at 10 kb, 30 kb, 60 kb, 100 kb, 150 kb, and finally 210 kb away from the anchor sequence. To test linkage at different distances, we extracted DNA from an immortalized B lymphocyte cell line and screened the DNA using these 7 mile marker duplex assays. This graph shows the percentage of linked molecules on the y-axis as a function of the distance separating the mile markers from the anchor sequence on the x-axis. Across 3 extractions, we found these data to fit an exponential model with a uniform DNA fragmentation probability per kb. Extrapolation suggests linkage can be measured out to approximately 300 kb in a single well. Our control for no linkage, which was comprised of an assay targeting a different chromosome, showed no linkage for any of the mile markers. Given we can confidently measure linkage out to 210 kb in a single well, we were curious as to how many genes are shorter than 210 kb in length in the human genome. Here are all the genes in the human genome sorted according to their length, as measured from the start to the stop codon. 94% of the genes are shorter than 210 kb, suggesting this method is sufficient for phasing variants in the majority of human genes. Now I want to spend a few slides on phasing variants using assays that cross-react. Here are 2 scatter plots. The one on the left shows data from 2 assays that are very specific. In contrast, the plot on the right shows data from assays that cross-react with the non-targeted allele. This cross-reactivity causes the droplets that contain the non-targeted alleles to fluoresce. In total, there can be as many as 16 discrete clusters. Fortunately, this cross-reactivity does not affect the ability to measure linkage using ddPCR as long as these clusters can be appropriately classified. As the color of the droplets suggests, the 4 clusters in each of the quadrants should be treated as if they are one population of droplets. Remember, digital PCR is a binary measurement. The software does not care about the absolute fluorescence of each droplet, but instead only cares whether the droplets are classified as positive or negative. By classifying according to the quadrant, these 16 clusters can be reduced down to the 4 conventional clusters shown here. Although in a duplex assay there may be as many as 16 clusters, not always will 16 clusters be observed due to differences in cross-reactivity, competition, and fluorescence between the assays. I will spend a moment discussing why cross-reacting assays create droplets of different fluorescent intensities so that you can properly classify your droplets and have confidence in your phasing data. Here I've illustrated a droplet containing a DNA molecule in which a lowercase a is the variant of interest. The FAM probe used in this assay matches perfectly with the sequence of this variant. During PCR, we expect this target to be amplified by the included primers during every round of thermocycling and the probe to land on the amplicon and experience cleavage, thereby separating the fluorophore from the quencher and allowing this droplet to progressively increase its fluorescence with every added cycle of PCR until the reagents become limiting. Remember, when phasing variants, the other allele is always present. In this illustration, the droplets contain the non-target allele, which are molecules that contain the uppercase A, are shown at the bottom. We expect the primers of the assay to amplify this target just as well as the molecules containing the lowercase a variant. We expect the total mass of PCR products between the droplets containing the targeted and non-targeted allele to be roughly equivalent. The only difference is a single base pair mismatch for the probe binding. This single mismatch significantly lowers the TM of the probe for the off-target molecule. As a result, at the annealing temperature of the reaction, only a small fraction of the probe that is specific to the lowercase a will bind to the uppercase A. This small fraction of probe does get cleaved and contributes to the fluorescence of the droplet, but only to a small degree, so that at the conclusion of PCR, these droplets have a low level of fluorescence. Remember, if a very specific assay was used, the droplets that contain the non-target allele would be indistinguishable from the negative droplets, but that is not the case here. At sufficiently high DNA loads, a third cluster of intermediate fluorescence intensity may be present due to cross-reactivity plus competition. This occurs when both targeted and non-targeted molecules are present in the same droplet. Because both are amplified equally well within the droplet and the total mass of PCR product within the droplet is limited, only half the PCR product will contain the perfect match for the probe. This causes less probe to be cleaved during every cycle of PCR and results in a droplet of mid-level fluorescence intensity. Now, looking at an actual scatter plot, I've labeled the clusters. Starting from the top of the plot, the PM alone cluster contains molecules for which the probe is a perfect match. The cluster labeled PM and MM is comprised of droplets that contain both the targeted and non-targeted allele. So there is a mix of amplicons that are a perfect match and mismatch for the included probe. Finally, the cluster labeled MM alone contains only non-targeted alleles, and as a result, the probe is a mismatch for the included amplicons, resulting in a low level of fluorescence for these droplets. This same pattern is present along the x-axis if the hex assay cross-reacts with the non-target allele. The identities of the molecules present in each cluster, including the middle clusters, can be deciphered by looking at the labels that are horizontally or vertically aligned with the cluster along each axis. If you apply this knowledge to interpreting the scatter plots you observe with your assays, you will be successful in using ddPCR to phase variants. Now let's review the three main applications for phasing variants. First, this methodology can be used to identify disease carriers versus those individuals who may be compound heterozygotes. As previously stated, this information is very important for genetic counseling and helping predict clinical outcome. Other applications include understanding allele-specific expression and mapping genomic structure. To summarize, I have shown that variants can be effectively phased in a single well. Using high-quality sample prep, we successfully measured linkage out to 210 kb in a single well. The methodology is site-specific and technically easy. Therefore, this approach does not require genotyping. Furthermore, this approach does not require genotyping the parents, which traditionally has been required to determine the phase of variants in a child. Also, this technology has a low cost per reaction, a fast turnaround time, does require long-range PCR, and is largely independent of PCR efficiency. I'd like to acknowledge that this work is the product of a collaborative team effort between Bio-Rad's Digital Biology Center and Harvard Medical School. From the Digital Biology Center, Tina Legler performed the majority of the wet bench work. Svilin, Niels, and Samantha contributed to assay design and statistical analysis. George, Sean, and Ryan were all crucial in pushing this application forward during its early stages of development. I'd also like to express my gratitude for the invaluable contributions in application development made by our Harvard collaborators, Nolan Kamataki, Catherine Wong, and Dr. Steve McCarroll. Thank you very much for your attention. I am happy to take questions.
Jack, thank you very much for that engaging talk. If you would like a copy of the QX200 ddPCR brochure, application notes, and publications list, they are available at the link to the left of your screen for download. If you would like any additional information, please feel free to reach out to your local Bio-Rad Droplet Digital PCR specialist as well. Jack will now take questions. Jack, our first question is regarding sample prep. What protocol do you use to isolate the genomic DNA for these long-range phasing assays?
Interesting question, Mohini. So not all sample preps are equal. We decided to test two sample preps which are commonly used in laboratories and are also amenable to automation. So we tested both silica columns and another magnetic bead-based chemistry. We found that silica columns actually shear the DNA pretty significantly, and we were unable to measure linkage beyond 60 kb. In contrast, with the magnetic particles, we were much more successful. However, the magnetic particles were not functionalized with silanol groups. Instead, it relied on a heavy weight polysaccharide to actually precipitate the DNA onto the surface of the beads. The actual name of the kit that we use is the PrepFiler Forensic DNA Extraction Kit. It's available from Life Tech. It works quite well. Not only, we don't believe it to be the only kit which will work. So depending on your application, you should really look to see if your current sample prep methodology is going to meet your needs for phasing your variants.
So our second question is regarding linkage. Did you try to measure linkage at distances greater than 210 kb?
Yes, we did. We realized that there are variants of interest, particularly like in the regulatory regions of genes that people might want to phase. So we designed assays farther out, specifically at 500 kb and also 1,000 kb from the anchor sequence. And we tried to measure linkage at these distances, and we were successful sometimes, but not successful other times. So we basically had inconsistent linkage measurements, and this isn't a reflection of the technology, just a reflection of the quality of the DNA that we had extracted. So the message we took away from this was if we wanted to go out at farther distances, we would really have to change our sample extraction technique to get more intact DNA.
So going back to sample input, what is the input of genomic DNA that you used? And then two, did you need to increase input for longer range SNP phasing? Great
question. So we've done some modeling, and we have found that the optimal DNA load for phasing is roughly 200 copies per microliter. That said, we have performed experiments where we included increasingly higher amounts of DNA, basically did a dilution series, and looked to see how reproducible linkage was across the dilution series. And we found it to be extremely robust across really the dynamic range of the instrument. But that said, if you want to get the most precise measurement, it happens to be at 200 copies per microliter, which is, of course, lower than the concentration you would use to get the most precise quantification of a single target.
So going back to measuring linkage, here's another question regarding that. Can you successfully measure linkage between more distant loci by merging wells together and performing meta-well analysis?
Yes, you can. So again, we've done some modeling here, and the benefit you gain from adding more wells is basically every time you double the number of droplets, you gain about a 10% improvement in the distance that you can successfully measure linkage. And again, this is an exponential function, so the returns are diminishing quite quickly as you get up to higher and higher numbers of merged wells. At a certain point, the experiment becomes quite expensive to perform, and you're actually much better off, again, rather than merging wells, changing your sample prep to ensure that you're putting higher quality DNA into your reaction.
So we have a question regarding Droplet Digital PCR Technology. How would this technology work as a replacement for FISH and CGH assays commonly used in genetic testing? And are there any cases in which you would not use ddPCR?
So let's see. Well, first looking at FISH. FISH generally is, to my understanding, used for genes, not necessarily for variants. So you could use FISH for sure to do some genomic arrangement studies, but FISH is not going to have the resolution that you would need to actually phase specific variants on a chromosome, just because a single probe is not going to fluoresce enough looking at like a metaphase spread of cells. So you really need digital PCR to phase variants, whereas if you were looking at gross genomic structure that's not based on variants but instead on conserved sequences, you could use FISH. ddPCR itself is much more scalable. It's not subjective, and it's going to be much lower cost if you're running through many samples. And for the same really applies also for CGH. Again, with CGH, you're going to likely lose any linkage measurement that is present just due to the way in which the technology works. I'm not sure exactly how you would do that. You would have to have two, specific hybrid oligos to an array and look for binding across those two spots, which I think would be difficult, but maybe somebody who's more of an expert at CGH would have other insights that I'm not seeing at this point.
So the approach you mentioned in your presentation, can this be used for somatic rearrangements?
So yes, it can. Somatic rearrangements, in contrast to the data we presented here, we presented just germline data, so 100% of the sample either had or did not have the variants. With somatic rearrangements, only a percentage of the sample is going to have the rearrangement. So the greater the percentage of the sample that has a rearrangement, the easier it will be to actually conclusively measure linkage and confirm that that rearrangement has occurred. Also, when doing this type of an experiment, it's easiest if the rearrangement event is very predictable, such that you can very carefully design your assays such that in the rearranged genomes, the two loci that you're actually querying are very, very close together, such that in the rearranged genomes, very little fragmentation exists to separate these two targets that you're looking to demonstrate linkage between. So it absolutely can be done. The sensitivity of the approach is really dependent on the percentage of the sample that is rearranged and how close these two targets are going to become in the rearranged sample.
Thank you for that. So in your presentation, it also appears that the fragmentation rate for the CFTR locus is linear, whereas the fragmentation rate for the mile marker locus is exponential. How do you explain this?
Yes, we were very perplexed by this at first glance because it definitely seems contradictory. And to make sure that we weren't missing anything, what we did is we went back to a CFTR cell line, we grew it up, and we did a mile marker study in the cell line. At the same time, we measured linkage between variants that were in the CFTR locus. And when we did that, and it was a single extraction, the two loci had fragmentation rates that essentially were equivalent. In other words, in this admittedly small experiment, we didn't see any distinct differences in different locations of the genome in regards to their tendency to become fragmented. And so we're looking back at our CFTR data, and again, it was across 6 different cell lines. Although the fragmentation rate was relatively consistent, there were differences among the cell lines, and we just think that it just so happened that it appears to be linear. But in fact, if we were to extend out and go beyond the 116, which was the shortest, it was the longest variance in these cell lines. If we were to go beyond that, you would really start observing more of that exponential pattern, which we believe is there, but was just sort of hidden in that data set.
So our last question for today is if troubleshooting related. So if we are having difficulty phasing variants in our lab, what should we do?
So my very first suggestion would be to do a mile marker experiment like I've presented here. You really need to get a good gauge for the quality of DNA of your sample. It may be that the DNA that you're working with is just so highly fragmented that the targets you're trying to assess linkage between are all busted up. And until you can conclusively show that the sample has enough intact, long molecules to definitively measure linkage, that should be your first step. If you are confident that you are measuring linkage at the distance that you're interested in, and when you go to phase variants it's just not working, it's possible that you're having issues with assay design. If that's the case, I would definitely recommend you contact Bio-Rad Tech Support. We have a great staff of scientists there to help you, and we can try to work with you to address your problem.
Thank you, Jack, and thank you everyone for attending the Bio-Rad Droplet Digital PCR webinar today. For those of you who had questions we were not able to get to today, someone from Bio-Rad will follow up with you in the next few weeks. For more information on Droplet Digital PCR and educational resources, visit us at biorad.com. Thanks, and have a great rest of the day.